Hey guys, this is the video for your exam review coming up on Tuesday. I am posting this on Sunday. Actually, I'm making this on Sunday because I made a video yesterday and tried to post it, and it would not post, and it kept telling me to contact customer service for YouTube, and turns out that I had music playing in the background, and I didn't even think about copywriting issues, and so they just told me to remake another one, so I'm remaking it today. So, with all that forward, your exam is on Tuesday, the 21st. Same place, same time, 12 to 1:15 in the musical theater building, in the same classroom that you have left for Zoom. This exam is going to require you to bring a Scantron. In the past, it's both been blue and green, doesn't matter which one. Just make sure you bring one that's uncrumpled up. Make sure it's nice and flat. You put it in a nice place in your book bag so when you come to class, it is still flat. Because when it is crumpled, it does not go through the scanner correctly. It either misses answers, it gets stuck, jammed, and it's a big mess. So just make sure you bring a nice, flat Scantron, blue or green. Make sure you bring a pencil. If you bring a pen, that can be used, but once again, if you mess up and you realize that you need to change an answer, you can't erase pen. So always best bet is to bring a pencil. So let's get started. As you can see on the syllabus, plus what Dr. Kokesh just went over in lectures, this exam is going to cover the introduction, the classification of microorganisms, the chemical principles, prokaryotes, eukaryotes, microbial growth, and microbial metabolism. I could tell you just from being VLA, this is my, oh goodness, fourth go around. So for the previous three, this exam is a lot of prokaryotes, eukaryotes, gram-positive, gram-negative. Of course, you're going to have to know introductions. So you're going to have to know some of the inventors, like Lester. Of course, we all know Pasteur, who is pasteurization. He also did the flask, which we all should know that by now. It's just know the inventors. I'm not going to say that you're going to see more than 3 questions, maybe 4, but you need to know them all because you'll never know which one you're going to get. In the past, of course, there's 2 main ones that you learn, Pasteur and was it Koch? You guys know what I'm talking about. I have actually seen exams where they don't even make an appearance, but ones like Lester do. So just know all the inventors. Prokaryotes and eukaryotes is going to make a huge impact. If you did concept map 1, that section where it says list the differences between prokaryotes and eukaryotes, that's what it's going to be. So it's going to be a lot of prokaryotes have no nucleus, eukaryotes do have a nucleus, eukaryotes have membrane-bound organelles, prokaryotes do not have membrane-bound organelles. The ribosomes, eukaryotes have 80S, prokaryotes have 70S. Then it's also going to get into a lot of gram-positive, gram-negative. So I would definitely know gram-positive, gram-negative, which one has a peptidoglycan layer, which one has the lipid layer, which one is blue and which one's red. So that's good stuff to know about. Those are pretty easy. Classifications is pretty easy as well. Just go over those, make sure you know those, make sure you have those together, because those are going to be a really important part of your exam. What I would say for prokaryotes and eukaryotes, since it is Sunday and you will have the rest of today and Monday and a little bit before your exam on Tuesday at noon, just make a list. The best part is to make a chart, a table, whatever you want, and just go back and forth. List the differences between prokaryotes and eukaryotes, and just do it over and over again. Don't just do it once and memorize that sheet, because yeah, that's going to be really good, and I'm sure it's going to help some, but the more and more you write it, the more and more you're going to remember it. So I would do it, look at it, give yourself a break. Always give yourself a break. Always walk away from it. So today, next week I have 2 exams, and I'm studying for them, but what I'm doing is I'm studying intermittently. So I'm not studying for, I'm in biodiversity right now, and I'm not studying all the clades, and I'm not studying all the trees, and then I'm studying math. I'm studying the clades, studying the trees, then I'm leaving, going to make dinner, going to the store, coming back, then studying math, and then coming back to it. It's always good because when you come back to it and you freshly write it out again, if you can do it all by memory the second time, when you do it the third, fourth, and fifth time, it's going to be easy. So when it comes to the exam, you're going to get that question and you're going to be like, oh man, okay. It's going to be like, in a prokaryote, which ribosome does it have? And Dr. Kokash could list 70, 80, and then make up ones like 60 and 40. She can also do the ones where it's going to say like, it is going to say 70S ribosome, 80S ribosome, nucleus, and I don't know, let's just say the endoplasmic reticulum. Let's just say that. And she's going to make you do the answer. So it's going to just say prokaryotes, and you're going to have to choose the one the prokaryotes have. And then it could be singles, or it could say use more than once. That's always a good option for microbiology. Another good option is true or false in microbiology. Does the prokaryotes have an 80S ribosome? Of course they don't, so you would choose false because they have a 70S. So it's always good just to know each type of question because in microbiology it can come out of nowhere. So you can have multiple choice with 4 to 5 answers and only one's correct. You can have true or false, which is, it's personally one of my favorites, but I know some that don't like it. And also matching. There's also when I took microbiology, it was a bunch of matching on my exams. So be ready for those kind of questions too. So that's what I would say for those. Same thing with gram-positive and gram-negative bacteria. Just go over it over and over. Write it. Take a break. Come back. Try to write it from memory. If you have to look at your notes the second time, that's great. But by the third time, try to do it all by memory. Because if you have it all by memory, then when you come to the exams and you see the questions, it should be fine. The two PowerPoints that I'm really going to focus on today is the microbial growth and the metabolism one. Because these are the ones that in the past 3 semesters that I've been the LA for Dr. Kokash, these are the PowerPoints that I have found that trouble the students the most. So I'm just going to go ahead and I'm going to pull out the material from the ones that I've either seen on previous exams or that I have a feeling might show up on your exam, and we're just going to go over it and talk through. I'm just going to go straight from the PowerPoints. If there's great figures that I see, I'm going to say, slide 55. This is the figure. Look at it because it's possibly. So I'm just trying to help you guys the best I can. Little tidbit though, I don't actually see the exams. So when you watch this video and you're like, well, is this going to be on the exam? I have no idea. I am not, as an LA, actually allowed to see the exams before. So I have no idea what Dr. Kokash can come up with. I can only go off of what I've seen in previous exams and what I saw in my exam when I took microbiology and just what I think is going to show up, only because it's important for the class, and not only important for the class, but important for your futures as whatever you're going to do as global health majors as nurses. So starting with microbial metabolism, I would definitely say look at slide 4. That's going to be good just to get your mind going, get your mind fresh on what this actually is. Definitely know catabolism and anabolism, which anabolism is the reactions that require energy for complete breakdown, and catabolism is reactions that release energy for ATP synthesis. It's always great to know the difference between catabolism and anabolism because not only we see this in microbiology, but if you go on to take chemistry classes, and these have actually showed up in my physics classes as well. So it's always good to know the difference between the two, and always it's good to know that metabolism is, of course, catabolism plus anabolism. We should all know by now that catalysts are the protein that catalyzes reactions. So it increases the rate, which means technically speeds it up, and it decreases the activation energy, which is the amount of collision energy required to start a reaction. figure, the figure on slide 8 is really good because I won't say you will see it on this exam, but I can promise you going forward in your career, you will see this slide again. I don't think I've ever seen Dr. Kokesh test on it, but if you want just to look over it just in case, because like I said, Dr. Kokesh can pull anything she wants. She doesn't ask me what I think. So if she wants to pull it, she can pull it. So it's always good to look over. But I would save this slide. If you have a folder, which I really suggest, putting a folder on your computer, save it in your documents, your desktop, because for your future careers. So as I'm going into master's school, starting in the fall, at Shenandoah for Applied Behavior Analysis, I actually have a folder since sophomore year that I kept important files in that I was like, man, I could use this in my future. And actually, classes from my sophomore year, I will be using in Applied Behavior Analysis that I kept. So I have the information. So this was a good slide to save if you're going on a future career in nursing or global health. I have seen these on slide 9, the enzyme components. So the haloenzyme is, of course, the apoenzyme plus the cofactor. The apoenzyme is the protein component, and the cofactor are the coenzyme. It's interchangeable names. Please know them both because she could throw coenzyme at you, and coenzyme is, of course, actually the cofactor is what the name is. So just know both interchangeably, and it's the non-protein component. So just remember the apoenzyme is the protein component, and the cofactor / coenzyme, whichever she uses, is going to be the non-protein component. And definitely know that haloenzyme is, of course, then plus together. Slide 10 actually has a great figure that shows the apoenzyme plus the coenzyme going together, and the substrate coming in to the apoenzyme and the coenzyme, and it's going to fit perfectly in there. And then, of course, substrates. Substrates are compounds that are acted upon as an enzyme, and they fit together as lock and key. So as you can see from the slide I just mentioned before, it's going to fit in perfectly between the apoenzyme and the cofactor because it's going to fit like a lock and key, like the lock to your front door, the key fits perfectly in. It's unique to it. And as you can see on slide 13, that is a greater illustration. The substrate comes into the active site. The active site is the site that actually is open for the substrate that can come in and fit like a lock and key. So you have the apoenzyme, which won't always look like a half-shaped moon, or I guess not, yeah, half-shaped moon, but most of the time it will. You will see the coenzyme, which is going to be a different color sitting near the top. The substrate is going to come in to the active site, fit like a lock and key. It's going to become the enzyme-substrate complex. It's going to sit there, products are going to go off, and then the substrate is going to release itself from the apoenzyme and coenzyme. There is definitely some influential factors to this, so I would definitely know slide 15. The temperature, of course, if it's increasing temperature, it's going to increase the rate. But of course, we all know if it gets too hot, it's going to denature the protein. So in the cold months, we love to have our house stay at 71, 72, some people 74. But if you leave your heat on too long and it starts, to get a little warmer, or just say the environment changes and your temperature goes up to 77, we're going to start sweating, we're going to get hot. Same with the protein. If it gets a little too hot, it is going to denature, which denature, of course, you figure out that it's just the unwinding of the protein, if you do not know that already. The pH, of course, the H concentration increases with decreasing pH. So please remember that. The H plus concentration is going to increase when the pH decreases. And of course, on slide 16, you can actually see what I was talking about. The active protein, it's intertwined. It's like on the color guard when they have the ribbons and they twirled. That's what an actual active protein looks like. It looks like the ribbon that is twirled. But when the temperature gets too hot, it's going to denature. And it's like when the color guard lays down the ribbon, it's just like a flat string. There's some neat graphs on here that you can see. And of course, we're right back to catabolism and anabolism. Catabolism generates energy, and the anabolism requires the energy. And that you can always look at The catabolism generates the energy because it's breaking down large molecules into small molecules. And then anabolism requires the energy because it's taking small molecules back into larger molecules. Okay, oxidation reduction reactions. If you've taken a chemistry class, you know this is a very essential component to the class. Same way with microbiology. There's a shorthand for this when it comes to oxidation reduction reactions. If you do not know it, it's oil rig. Oxidation is losing. Rig. Reduction is gaining. That is going to be an essential tool. I would write it on the top of your exam, and then I would say oil rig, and then bring it down. Oxidation is losing. Reduction is gaining. This is going to be an essential part of the transfer of electrons. So as you can see on slide 22, as I was just reiterating with the analogy oil rig, oxidation is the loss or the removal of electrons. So the electron donor then becomes oxidized because it lost electrons. Reduction is the gain of an electron, and the electron acceptor becomes reduced because it just accepted or gained an electron. And oxidation and reduction reactions must be coupled. There cannot be a free-floating electron. That is not a thing. It does not exist. So if you have oxidation, you're going to have reduction at the end because it cannot be floating. So it comes in a coupled pair. They're always together, and you will never see them separate. Slide 23 actually gives you a good point of the oxidation reduction reaction. It gives you a great example. So I'm almost positive this will come on the exam. So please study 23, where A loses an electron and B picks up that electron. So A becomes oxidized, and the electron donor is now oxidized, and B has been reduced, so the electron acceptor has been reduced because it gained an electron. And there is a great visual, so if you're a visual learner, it is great on slides 24 and 25. It gives you a great visual aspect of what slide 23 did in verbal language. ATP. Okay. This is going to show up for the rest of the class until exam 3, when we start getting into the actual diseases. But for exam 1 and exam 2, ATP is going to be your best friend. ATP is a major molecule that traps and carries the energy released in catabolic reactions. So there are 3 ways that organisms form ATP. 1, substrate level phosphorylation, 2, oxidative phosphorylation, and 3, photophosphorylation. 1, substrate level. Substrate level is the direct transfer of high energy of PO4 to ADP. So then the phosphenyl pyruvate plus ADP then turns into pyruvate plus ATP. So pretty much it's just PO4 transfer the high energy to ADP. Then so that now turns it into PO4 goes to pyruvate and ADP goes to ATP. And like Dr. Kogash does mostly on slide 28, if you're a visual learner, there's a visual aspect of adding the energy transfer from PO4 and ADP to pyruvate ATP on slide 28. 29 is oxidative phosphorylation. This involves the ETC, better known as the electron transport chain. And it just takes NAD plus to NADH and NADP to NADPH. The ETC happens at different places depending on if it's a prokaryote or eukaryote. Prokaryotes happen in the plasma membrane, and the eukaryotes happen in the mitochondrial inner membrane. So just that's another difference between prokaryotes and eukaryotes. That could be asked in the exam, and you should add to the charts that you make from the information you got from the prokaryotes, eukaryotes PowerPoints. That the prokaryotes have the ETC in the plasma membrane. Yeah, the prokaryotes have it in the plasma membrane, and the eukaryotes have it in the mitochondrial inner membrane. Also know for oxidative phosphorylation that the final electron acceptor is going to be oxygen. And then 3, the third way to get ATP is photophosphorylation. This is only in photosynthetic cells. Oh my goodness, early in the morning. and that's usually cyanobacter, are the green plants algae. Um, and of course, end result, we all know what photosynthesis is by now from previous biology classes we've taken. The end result is just ATP from sunlight. Slide 32 is also going to be a big friend of yours. It's going to talk about the aerobic respiration, the fermentation, and the anaerobic respiration. Definitely know slide 32. That will show up in your exam. I'm almost 110% positive. Study slide 32 in the microbial metabolism. Also know glycolysis. Of course, glycolysis is the splitting of sugars. It's a series of 10 reactions where glucose is oxidized into 2 molecules of pyruvic acid. And of course, during glycolysis, NAD is reduced to NADH, 2 ATPs are produced, and there's no requirement of oxygen, of course, because it is glycolysis. So the net ATP gain from glycolysis is going to be 2 ATP. Please remember these values because they will show up. I'm almost positive there's going to be questions that's going to ask you, what are the values of ATPs? What are the a very, very good study tool is if you did concept map 2, please go back to the tree. If you did not do the tree correctly, that is fine. It's all been corrected for you. But now you have the correct answers. If you did it correctly, great. Please study this. Please study this because that is going to be a great tool for you along with the PowerPoints. There's great illustrations. So Dr. Kokash always provides you guys with visual illustrations because there's a technique that she learned, of course, while becoming a professor, and what I learned while I was becoming an LA, and for the past 4 semesters that I've talked to the new LAs and us returning LAs that help the new LAs, that there's always different kinds of learners. Their students always come to them and say, I really don't understand visual text, and Dr. Kokash understands that, so she always provides you guys with visual illustrations. And of course, these pathways are ridiculously hard, just reading the words, so she always gives you visual illustrations. Please go to, starting on slide 35, it says 36, 37, 38. Those are all great. And 39 as well. Always go to look at these because if you do not understand written words, which I have a time and struggle learning written words when I am doing any class, and microbiology is one of the important classes you will take, please go and look at these pictures. These pictures are great. They go through each detail and step very accurately, very simply. They have great words and great text of what exactly is happening in this photo. Please go look at these photos if you are having any trouble with these pathways at all. And the same with, of course, the Krebs cycle. Of course, anything to do with the electron transport chain. Just go over these. Just go over them in excruciating details. I know we all have important things happening in our lives right now, but carve out some time. Carve out some time tonight. Carve out some time tomorrow. Wake up early on Tuesday, but please make sure you get a good night's rest on Monday night. Wake up a little early on Tuesday and really go into this with excruciating detail, because the more detail and the more time you go on studying, the better we've noticed you do on the exams. This is not a class that you can just start studying Tuesday morning and ace the exam. I can guarantee you that right now. So please study tonight, study tomorrow, and hopefully you guys have started studying. This class requires you to study a lot. I used to study about a week ahead, and I know some students that got pretty good grades in this class that have told me they started studying about a week before the exams too. So if you do not do it this time, study really hard. Try to do well. But just remember, Dr. Kokash does drop one exam, so if you don't do so well on this exam, just remember that she does drop one exam and that your final now counts as 2. And then start studying for exam 2, 3, and the final extremely hard. Like, harder than you've ever studied for another exam before, because this class is hard. I'm not going to sugarcoat it, and I know Dr. Kokash hasn't sugarcoated either. This class is extremely difficult, but it's extremely knowledgeable, and you will use this information going forward. I promise you that. I mean, I've even found a few aspects of microbiology in applied behavior analysis, which applied behavior analysis is, in simple hand terms, if you've ever seen Criminal Minds, their behavior analysis, that's what I'm going to do, but not on a government FBI level. I'm going to use it for the military with PTSD, but I've even found aspects of microbiology in my career. So I can guarantee you will find a lot more in your career. So that's pretty much the microbial metabolism. Study it in excruciating detail. That's all I can say. Yeah. If my video has been freezing, which I see it is now, so if you see me doing crazy movements, I'm sorry. It's just going really slow, and I don't really understand why. So microbial growth. Once again, this is pretty much just know every word in the PowerPoint. But just go over a few. Obligate and facultative. Obligate, of course, is a specific environment, and facultative is able to adjust to its environment. There's, of course, physical requirements, pH, temperature, and osmotic pressure. There's biochemical reactions, which is seen in sources, which we all know is carbon and nitrogen, H2O, which we all know is water, and oxygen requirements, as well as organic growth factors and trace materials. I've seen this pop up on exams, which is the temperature, psychrophiles, which is loving, mesophiles, which is moderate temperature, which we are mesophiles, and thermophiles, which is heat-loving, that are about 40 to 80 degrees Celsius. I did have a question on my exam that said, which one of these would be great in a hot spring? And it was an extreme thermophile. So if that does pop up, you got a leg up. It's extreme thermophile and hot springs, because hot springs are ridiculously hot, of course, by its name. Just know that, of course, the pH, the osmotic pressures. This also will follow you chemistry. I've seen it in physics, and it will follow in other biology classes. The osmotic pressure: hypertonic, hypotonic, and isotonic. Isotonic is neutral. The same goes in as the same goes out. It stays the same. Hypertonic, on the other hand, is the solution concentrates of solutes outside. So everything goes outside, which means it actually shrinks. And of course, hypotonic, the solution goes inside of the cell and it bursts. So just remember, it's hyper. Hyper is getting skinny and shrinking. Hypo is getting all of it inside. It actually is about to burst. There's a great illustration of this on slide 8 if you need a visual requirement. Heterotrophs and autotrophs. I know this is also a big deal. This was also on Concept Map 2, so please go study that. If you got it wrong, it was corrected. If you got it right, it's still right. Heterotrophs are, of course, carbon is derived from organic compounds. You need that. And that's proteins and carbohydrates. Autotrophs is carbon from CO2, which is, there's two different breakdowns. Chemoautotrophs get their energy from, of course, organic materials, and photoautotrophs, of course, like photosynthesis, get theirs from sunlight. It goes down into the different elements: nitrogen, sulfur, phosphorus. I would know a little bit about each. I would definitely know about nitrogen fixation. Then oxygen, of course. We have the different levels. So it's going to be obligate anaerobes, aerobes, aerotolerant aerobes. There's a great illustration of this on slide 17. But just a quick, review. Aerobes, of course, love oxygen. Anaerobes do not love oxygen. Anaerobes cannot be in the presence of oxygen. Nothing will grow. Same with different from that is aerobes, which love oxygen, and if it's placed in an environment with no oxygen, nothing will grow. There's a few different ones. Also, obligate anaerobes, which only anaerobic growth ceases in presence of oxygen. We all know that. Then there's aerotolerant anaerobes, which is only anaerobic growth, but continues in the presence of oxygen, and microaerophils, which only aerobic growth. Oxygen is required in low concentrations. So it's 5 different ones. Obligate aerobes, which of course oxygen is absolutely 100% required. Facultative anaerobes, which is both aerobic and anaerobic. So there's greater growth in the presence of oxygen, but it can grow it without oxygen. Obligate anaerobes, which is absolutely no oxygen present. Here's a little tidbit. If it's obligate aerobes, needs oxygen. Obligate anaerobes doesn't need oxygen, will die in the presence of it. Those are the two. If you see obligate, that's when you know. The ones that say facultative, micro, and aerotolerant, that's when it gets a little sketchy. So once again, the facultative anaerobes is both aerobic and anaerobic, but it grows greater in presence of oxygen. Aerotolerant, only anaerobic growth, but it will continue. So aerotolerant anaerobes, even though it's anaerobic, it will continue to grow in presence of oxygen. And then there's microaerophils, which is only aerobic growth, but oxygen is only required in very low concentrations. So like I said, if you don't really get it when it comes to visual text or me speaking to you auditorily, please go to slide 17, you'll get it visually. Biofilms. Also go over the biofilms that will show up on your exam. So please know what biofilms are and what they're composed of mainly. What they do, so like they share nutrients and they also shelter each other from harmful factors. So just know little tidbits of biofilms and what they exactly are. Also know the generation or doubling time. So the length of time in which a generation of cells is divided, which of course when cells divide, of course they're going to double in numbers. Know that it depends on what particular organism it is and what the environmental factors around it is. Is the temperature too hot? Is the temperature too cold? Or is the temperature just right? Also know the growth curve. Now, I could explain it to you up, down, left, sideways. The 4 different stages of the bacterial growth curve is lag, log, stationary, and death. But I have seen in previous exams where you actually have to draw it and label it, or it's already drawn for you and you have to label it. Please go to slide 35. Know what the stages are, lag, log, stationary, and death, but please know how to label it, where it starts, where it ends, and what each stage is when it comes to the graph that's going to start lagging, logs going up, stationary, we all know, stationary, then it becomes now flat, and of course we all know death is down. So I'm expecting you all to get 100% on this question. Just remember, lag, you're lagging. You're not really going nowhere. You're holding behind. Log, you're like, okay, now I'm starting to pick up a pace. Stationary, you get tired and go flat again. Death, you're done. I'm not running anymore, I'm out. So please remember that. Please know where each begins and how to label it. Slide 35 and 36. 36 is a little bit more busy. But if you guys like busier slides and visual aspects, that's great. Slide 35 is standard. It's literally the graph, and it shows you where to label it. Please know where to label this graph. That's it, guys. That's what I got for you. I wanted to go over those two mainly because those are the ones that I see people have struggles with the most. But yeah. I'm looking forward to you guys doing well in this exam. I know you all have it. It's gonna get nothing harder over here, folks. So study, study, study. Like I said, I started studying for mine a week ahead, and I know the students that got really good grades in this class started about a week before the exam. So for exam 2, 3, and the final, please start studying about a week ahead. But yeah. Just remember, your exam is on Tuesday the 21st, which is in 2 days. Same place, business, building, oh my gosh, I'm so sorry. Building, classroom, and time as lecture with Dr. Kokash, 12 to 1:15, musical theater building, in the same classroom number you guys are in now. Remember, bring a Scantron, it's green or blue, doesn't matter. Just make sure it is not crumpled up and it's nice. And bring a pencil. Other than that, good luck, guys. I know you all have it. See you for the next one.